Welcome everybody to the 3D Regulatory Genome, our seminar series, virtual seminar series here at Enhanced 3D Genomics. We are starting 2025 with a very exciting speaker, Dr. Evgeny Kvon, who I've only just met in person for the first time 5 minutes ago virtually, but whose work I've followed and admired for a long time. And I've admired both the conceptual advances that he's contributed to, but also the technology development. And I think we'll hear both of this during today's seminar. So let me just share. This is today's speaker. I'll give a very brief background, so the housekeeping. If you have any questions, if you could please put them in the Q&A box and I'll read them out at the end of the talk. And the talk will be recorded, but not the questions, not the discussion, right? So that's it. And with that, I'll welcome Evgeny. Evgeny obtained his PhD from the University of Vienna, where he worked at the IMP in the lab of Alex Stark, who is, of course, very well known in the enhancer field, and then went to do his postdoctoral training in the labs of Len Pinacchio, Axel Wieser, Diane Dickel, and from there moved on to his current position, where he is at the Department of Developmental and Cell Biology at UC Irvine, where he's an assistant professor and is studying how genes are regulated during mammalian development by long-range enhancers and how their misregulation leads to congenital disorders. Very much looking forward to your talk, Evgeny. Stage is yours.
All right. So thank you, Stefan. And I'm really honored to be here as I think one of the first speakers, right, of the seminar series. And so I'm going to tell you a little bit about our recent work on understanding how genes are regulated during development by enhancers, and also partially cover how variants in enhancers cause congenital disease. So during development, a single cell, which is fertilized egg, gives rise to a complex organism with many different cell types. So all of these cell types have identical genomes, yet they have all different functions, right? And this is due to differential gene expression, right? Because neurons express neurotransmitters, blood cells express hemoglobin, etc., right? And this differential gene expression is a main reason why every cell in our organism has its unique identity. One of the major regulators of gene activity are enhancers. Enhancers are regulatory non-coding DNA elements that are somewhere between 100 and 2,000 base pairs in length that contain binding sites for transcription factors. And upon binding of these transcription factors, enhancer loops to the target gene and activates it in a spatiotemporal specific manner. For example, here in the mouse brain of this developing embryo. A typical mammalian gene contains many such tissue-specific enhancer elements. For example, this hypothetical gene here has two enhancers, one which is active in the brain and another that is active in the limb. And the combined action of these two enhancers results in a more complex gene expression pattern in the limb and in the brain. So one of the challenging things about studying enhancers is that they are located in a non-coding part of the genome, which is actually the largest part of our genome. 98% of our genome is non-coding. And so by some recent estimates from the ENCODE project, a human genome contains probably several million enhancers. So they far outnumber the genes in our genome. And another feature of mammalian enhancers is that sometimes they act from a very long distances, sometimes up to several million base pairs. So my lab is interested in two kind of big questions about enhancers. So the first question, which is one of the biggest questions in the field, is how do they work remotely, right? So as I mentioned, many enhancers allocated millions of base pairs away from the promoters, but so how do they specifically activate the target genes is still unclear. And I'm going to cover two of our recent studies where we assess an importance of enhancer promoter looping in developmental gene activation, and another study where we uncover a new kind of cis-regulatory DNA element that seems to be critical for mediating long-range enhancer activation. And then the second smaller part of my talk, I'm gonna address maybe more disease-related questions. How do we actually functionally test variants that occur in enhancers in vivo? And so this is especially relevant because, as you all know, most of the candidate disease-associated variants coming from GWAS and whole genome sequencing studies affect non-coding DNA, and presumably many of them affect enhancers. So, okay, so I'm going to start with the first question. So how important is enhancer promoter physical proximity for gene activation? So this seems like a trivial question, right? Because if you look at the textbook, the definition of enhancer is that upon activation, it loops to the target promoter and that activates it. And then when it's inactive, it's not looping. And work on, from many labs over the last 30 years, for example, at the beta globin locus showed that it is indeed the case. And also, we can artificially force enhancer to the promoter, and it will actually cause its activation, as was shown beautifully by Gerd Lobel lab. However, there are some studies, especially in the early Drosophila embryos and mouse Hox locus, where enhancer seems to be proximal to the promoter irrespective of its activity status. So if it's active, it's close. If it's inactive, it's also close. And perhaps more counterintuitively, relatively recent studies on sonic hedgehog and SOX2 neuronal enhancers show that upon activation, enhancer actually moves further away from the target gene. So which of these models is correct, right? Yeah, and so the kind of difficulty of deriving our kind of generalized model is that only few bona fide mammalian enhancers have been studied with high resolution methods, right, to assess the looping. And there are a lot of genome-wide studies, but they often use H3K27 acetylation or open chromatin as a proxy for enhancer activity. And those regions, a lot of them are actually not enhancers, and they're only predictive of enhancer activity. And to exemplify this is a typical method that is used in mouse embryos to identify putative enhancers, where we isolate the tissue of interest from a mouse of embryo, and then we can perform ChIP-seq with enhancer-associated histone mark, such as H3K27 acetylation or P300. And then you do a pull-down, you sequence, and then it results in a genome-wide map of enhancers, right? So this is a really useful method to derive a genome-wide map of putative enhancers in a single experiment. And then, however, if you want to say that the region is an enhancer, you need to validate it, right? So, in a typical way it's done is you take that putative enhancer region, you put it next to the reporter gene, in this case LAG-Z, and then you make a transgenic animal. And if enhancer is indeed an enhancer, it's able to enhance this reporter gene, and then you see activity in the mouse embryo. In this case, it's a limb-specific enhancer, right? This is operational definition of enhancer that was introduced by Walter Schaffner and others in the beginning of '80s. And so if you do this exercise, only a small fraction of this genome-wide of this H3K20 constellation peaks actually driving activity. So you have to do validation, and in cells you can do MP arrays, etc. So we wanted to kind of use this experimentally validated enhancers to ask a question about enhancer promoter looping. And specifically, we want to ask, okay, you have a mouse embryo, you have this brain enhancer, midbrain enhancer. When it's active, in active state, does it loop to the promoter or not? And conversely, in other tissues where enhancer is not active, whether it's also proximal to the promoter and not. And we want to test it for many enhancers, right? That were experimentally validated. So lucky for us, Axel Wiesel and Len Pinacchio, my postdoctoral advisor at Berkeley Lab, developed a resource of experimentally validated mouse and human enhancers. And they were validated through transgenic reporter assay one by one. So each of this almost 2,000 enhancer was cloned individually in front of the minimal promoter and the LAG-Z reporter, injected in a mouse egg, and the activity was assessed in E11.5 embryos. So this was a lot of work over the last more than 20 years. So, and then this database contains enhancers that are active in all different tissues of mouse embryo, including brain, limb, neural tube, etc. So we wanted to use this resource to kind of study what's a 3D conformation of this experimentally validated mouse enhancers. And of course, the method of choice is to do Hi-C, right? If you want to do the Hi-C throughput. And briefly, for people who don't know about Hi-C, you cross-link the DNA, and which preserves this 3D loops between different genomic regions, and then you cut it with restriction digest, you biattenulate, you ligate, and then you pull down this biattenulated ligation junctions, and then you sequence them. And this results in this genome-wide contact matrix that you see in the papers. So it's great to assess genome-wide 3D chromosome conformation, but for mapping enhancer promoter interactions, it's not typically you need to sequence really deep. But there is a there is a resolution problem if you want to study enhancer promoter interactions, and enhancers, as I told you, are very short, right? So solution which many people in the field use is to use capture, right? So you can do a Hi-C library, and then you can use oligos against regions of interest, and then you can just sequence regions that contact your regions of interest by using capture and bite and pull down. And so we did exactly that. So we, a postdoc in the lab, Zhuo Xin, dissected a mid-gestation mouse embryo in 10 different tissues, forebrain, midbrain, neural tube, face, limbs, etc., and prepared Hi-C libraries from each of these tissues. And then she used probes against around 1,000 of these experimentally verified VISTA enhancers, some promoters, and also some control regions. And then she sequenced these libraries. And then what it essentially results in is you get tissue-specific enhancer contact maps for this 1,000 developmental enhancers. And so this is how the data looks like. So you have, in this case, a brain enhancer that is located in the middle of the gene desert, but which of the genes around is actually a target gene, right? So with the capture Hi-C gives you a lot of these tracks that looks like 4C essentially, right? So you have this interaction profile, you have a lot of interaction close to the bait region. In this case, it's an enhancer. And then you can see that there is a peak around Zik1 and Zik4 genes, suggesting that those two genes could be target genes. And indeed, if you look at the expression of those genes, it matches enhancer activity, suggesting that it could be indeed the target genes for this HS654 enhancer. So, but we not only get it for one enhancer, we get it for all 1,000 enhancers across 10 different tissues. And so this is just a summary of the interactions. Most of them, of course, occur within TAD, like around 8%, and they're all kind of different interactions, including enhancer promoter, enhancer CTCF, etc. So we also experimentally validated some of these interactions by performing enhancer knockouts. In this case, as I already told you, HS654 enhancer is predicted to interact with Zik1, Zik4 gene. So we deleted it from the mouse, and then when we performed RNA-seq from midbrains and compared enhancer knockout versus wild-type mice, we can see that the only significantly downregulated gene is Zik4. Zik1 is downregulated, but it's not significant. And this validates the interaction that we predicted with capture high C. And here's another example. There's a cluster of brain enhancers that are contacting miR9-2 gene and across the distance of almost 1 megabase. And then when we delete them, indeed, the most significantly downregulated gene is miR9-2. Importantly, this gene in the middle, which does not contact these three enhancers, even though it's located closer to them, is not changing, validating the specificity of the capture high C interaction. And so this scenario when enhancers bypass their immediate neighbor gene is actually pretty common. In fact, more than 60% of all enhancers that we assessed do not regulate their immediate neighboring gene. They bypass their neighboring gene to regulate a more distal gene. And of course, a big unanswered question in the field is how is this specificity achieved? And so we looked at the skip genes and tried to see any evidence of, repression, a polycomb, etc. We couldn't see anything. We also looked at, different promoter types, but, the skip genes were not different from target genes. So, but the only thing that stand out was that the skipped promoters, half of them were fully methylated at the CpG, at the promoters, and they were also repressed. They were not active, right? So that's kind of suggests that this is I have to say a pure speculation that in this 50% of cases when the bypass promoter is methylated, methylation could in principle kind of maintain that skipping status, right? And then there is actually evidence for that from a paper from Moonwas lab where they showed that if you relocate that methylated repressed promoter in the middle, just 10 kilobases away, it will get reactivated by this same enhancer. So it could be that in this 50% of cases, methylation does prevent activation by this close enhancer. Of course, this is pure speculation, and what initially establishes that specificity is still unclear. It could be also not methylation, but something else. So, but now going back to this question that we wanted to address, okay, is looping important, right? So now we can actually address it for a lot of enhancers that were experimentally validated. And so in this case of this HS654 enhancer that I showed you before, if you look at contact profile of this enhancer across 10 different tissues, you can see that the strongest interactions are formed in tissues where enhancer is active, in brain, neural tube, and tail and trunk. But in tissues where, in tissues where enhancer is inactive, there's no contact. And so we validated it with fish, the super resolution fish, and I did see that in tissues where enhancer is active, they're closer together than in tissues where it's inactive. Mind you, this is measured across the entire population of nuclei. So the difference here is not as strong as we see in capture high-C, because capture high-C is based on cross-linking, right? And we're looking at just a small population of cross-linked nuclei. Where here we're looking at the entire population of nuclei. So there is a significant difference, but it's not as big. And now we can, if you look globally at enhancer promoter interaction intensity and cluster it by tissue type, we can see that there are some enhancers that preferentially contact the promoters in the face, in the limb, and heart, in the brain, etc. And if we now take that heat map and make the same heat map with actual enhancer activity, so this is a completely orthogonal data set, right? So this is interaction frequency based on capture high-C. This is enhancer activity based on a transgenic reporter experiment, which is a completely different type of experiment, right? And you can see that those heat maps kind of mirror each other, right? That the contact map mirrors activity map. And if you do a meta plot, you can see that indeed the enhancing promoter interaction frequency is highest in tissues where enhancer is actually active based on a transgenic reporter assay. So take home messages for this first part. So 60% of developmental enhancers that are long range bypass the neighboring genes. And so we could actually measure it now for experimental validated enhancers. And it seems to be linked to CpG methylation, which might or might not contribute to this skipping. And so most of the mammalian developmental enhancer promoter interactions are tissue specific. We also did some analysis of enhancer interactions, but I'm not going to talk about it. It's in the paper. And what I also didn't talk about, there's a smaller fraction of interactions between enhancer and promoter that in our assay look invariant. So they're always contacting the promoter irrespective of activity, but it seems to be forming by neighboring CTC outside. So what we think is going on, and what people have seen in other systems, is that enhancer might be tissue specific, but the CTCF site might make it look like a stable loop. So, and I want to end on there is this model from Wendy Bickmore that that suggests this zone of influence, right? So basically what the model proposes is that enhancers and promoters enhancers can activate promoter only within a certain zone of influence, which is I think around 400 to 500 nanometers, if I recall correctly. If you're outside of that zone, activation cannot happen. If you're within that zone, activation can happen, but it doesn't have to happen, right? It's just kind of a zone of competence. So, and that kind of explains this discrepancy between capture high-C studies and fish studies, where they see sometimes if you do fish, that enhancer actually goes further away from the promoter, but it's still within that zone of influence, right? And the zone of influence is what we detect in the high-C experiments. So now I want to talk about a different question, which is also related to long-range activation. And the question is, how do they actually do it, right? So for short-range enhancers that are located close to their target promoters, for example, in this case, it's PTF1A pancreatic enhancer, located only 25 kb from its target gene. The activation can happen just because the enhancer is actually close to the gene, right? But there are some examples of enhancers that are located really far away. And my favorite example is this brain enhancer of MYC that is located almost 2 million base pairs from its target gene MYC, still within the same TAD, right? So how does this work, right? And of course, work by many, many labs showed an importance of higher order chromatin organization. So we know that our genome is partitioned in so-called topological associated domains that are separated by boundary elements, and enhancers can activate genes only within the TADs, but not outside them. So the TADs can provide that rigid structure that restricts the zone of action of enhancers, right? But then there's also this maybe a little controversial work suggesting that if you remove, if you transiently remove cohesin, CTCF, from cells, there are actually very little gene expression changes. And in fact, most of the enhancer promoter interactions on a short time scale are maintained. And also, many enhancers can bypass CTCF boundaries. So which is also suggesting that enhancer promoter interactions, can are stronger, right? So what all this data suggests is that enhancer promoter affinity might be important for maintaining this enhancer promoter interactions, right? But what these additional genetic factors that regulate this long-range enhancer promoter interactions is still unclear. So, and as I showed you with the transgenic reporter assay, if you want to find those additional factors that affect long-range enhancer activity, the functional assay that we use to test enhancers doesn't really address that. And the reason why is because we force enhancer directly upstream of the promoter, right? And this reporter assay only assesses short-range enhancer activity, right? It doesn't tell us anything about long-range activity. So, and ideally, we want to develop a functional assay where we have remote enhancer located hundreds of kilobases from a target gene, where we can swap other enhancers and see if they work, right? So this is kind of like a long-range enhancer reporter assay, right? And in this case, we will be able to assess, okay, if that enhancer works long-range or it doesn't work. The caveat here is that most long-range developmental enhancers have shadow enhancers, right? And as the enhancer field, both in mammals and in invertebrates, learned painfully that if you delete the enhancer, very often nothing happens because there is another enhancer that is redundant, right? But there is one notable exception, which is a famous ZRS enhancer of Sonic Hedgehog, which activates ZRS activates its target gene over a distance of 850 kb, and it does it in a specific region of posterior limb bud here, right? And this is shown mouse limb here, skeletal stain of mouse limb. So this enhancer is actually important for Sonic Hedgehog expression in the limb. If you delete it, there is no expression of Sonic Hedgehog in the limb bud, and there's no distal limb outgrowth. So it's critical for limb development and for Sonic Hedgehog expression in the limb. So this is an ideal system where we can now test long-range enhancer activity. And to do that, we chose 4 very strong limb enhancers that are active in the same cell population as ZRS, namely posterior limb bud. All of these enhancers are open in the relevant cell types. They're conserved, and they're marked by H3K27 acetylation. So they have all the bona fide features of strong enhancers, and they drive strong LAG-Z expression in the reporter assay. So a very talented grad student in the lab, Grace, she created a series of knock-in mice where we replaced ZRS with each of these 4 enhancers one by one. And surprisingly, none of these enhancer swaps could trigger any Sonic Hedgehog expression from that remote location. And consistent with the lack of the Sonic Hedgehog expression, all these mice had truncated limb phenotypes indistinguishable from complete loss of function of the ZRS in the limb. So what it means is that enhancers are not simply interchangeable at a remote location, like they are in the transgenic reporter assay, right? So what's going on? So to cut the long story short, we ruled out a lot of possibilities. So the first thing we looked at was, of course, enhancer promoter incompatibility. So when we test enhancers in a short range assay, we use a different promoter. And so we tested it with a Sonic Hedgehog promoter. It still worked. And moreover, we also inserted one of these enhancers directly upstream of Sonic Hedgehog in the native locus, and it also caused its activation. So it's not enhancer promoter incompatibility. We also ruled out enhancer silencing, right? So we thought, okay, we knock in some ectopic enhancer where it's not supposed to be. So it could be that it's just getting silenced. And so we performed a TAC-seq and confirmed that enhancer is still open at the transplanted location, but yet it's unable to activate our long range. Okay, so perhaps there is something else, right? And when we looked at the enhancer promoter distances, but at the native enhancer loci, what we saw that most of these 4 enhancers are actually at a much shorter distance from their native target genes than ZRS Sonic Hedgehog, right? So as I told you, ZRS Sonic Hedgehog is 850 kb away from each other in the mouse. These 4 enhancers are at a much shorter range from their target genes, right? However, so our hypothesis here was that, okay, maybe these transplanted enhancers are intrinsically short range and unable to work over long genomic distance. But this was still kind of what didn't fit in this hypothesis was that if you look at this SAL1 limb enhancer called HS72, it's still located 410 kb from its target gene, right? This is not a short range by any means, but it still doesn't work, right? And so when we inspected the sequence of that enhancer in more detail, this is the orange bar shows the sequence that we assessed in a short range assay and that was swapped and couldn't rescue limb development. It's located in this, there's a very highly conserved sequence in the middle of that enhancer, but there is this also a very highly conserved sequence that was cut, but was not included in this minimal enhancer region. And so we hypothesized maybe that sequence is actually important, right? But it's important not for short range enhancer activity, but for long range activity. And so that additional element, which we later called range extender, maybe is needed to confer longer range activity. And to test that hypothesis, we created another knock-in mouse where we replaced ZRS with an extended version of HS72 that contains this range extender element. And to remind you, just enhancer on its own is not enough to activate Sonic Hedgehog. But when we added this 200 base pair sequence, it was enough to restore Sonic Hedgehog expression in the limb bud and resulted in fully developed limb that actually also had polydactyly due to broader activity of HS72 enhancer in the limb. Importantly, this range extender element is not just another enhancer, right? So if you put it in front of the minimal promoter and LAG-Z, it doesn't drive any expression. So it's not just adding another enhancer, right? And so the next question we ask, okay, it seems to be required for this HS72 enhancer, but can it convert another short-range enhancer into a long-range enhancer. And to do that, to test that, we chose the shortest range enhancer we had, which was enhancer of PRDM16, located 71 kb from its target gene. And to remind you, replacing ZRS with that short-range enhancer was not able to start Sonic Hedgehog expression. When we added the Rex element, it rescued Sonic Hedgehog expression and resulted in a fully developed limb, suggesting that not only Rex element is required for long-range activity, but it's also sufficient to grant long-range activity to another completely different short-range enhancer, right? And so what's remarkable here, we extend the range of this enhancer by an order of magnitude, right? From its 71 kb to 850 kb by just adding this short sequence element. Okay, so of course, the next question is, what is in the sequence of that range extender that is important and that makes that Rex element work, right? So we checked, of course, the first thing, there was no even weak CTCF site in that element, right? So, and also nowhere nearby. CTCF is not involved here. But we found a couple of very conserved transcription factor sites, a couple of which brought our attention. So there were two binding sites for transcription factors called LHX2 and LHX9, and one binding site for transcription factor called LEF1. And so to test if any of these motifs are actually important, we made another knock-in mouse where we now have a range extender with mutated LEF1 motif next to HS72 enhancer. In this case, this knock-in mice resulted in fully developed limbs, suggesting that this LEF1 site is not critical for range extender activity. If you remove it, it still works. However, when we removed both LHX sites, this range extender element lost its function. And it resulted in a mouse that had truncated limbs, right? So this was indistinguishable from just not having the Rex element at all, right? So that suggests that this LHX motifs are critical for range extender function. And so, okay, are these motifs present also in other long-range enhancers, right? And so when we looked at experimentally validated and predicted limb enhancers from our previous study and from other studies, and when we asked, are there sequence motifs that are enriched in long-range enhancers that are 400 kb and longer versus short-range enhancers? And in both cases, the top enriched motif was LHX2 and LHX9. And moreover, the presence of this motif correlates with enhancer promoter distance. So if you have more than two or more of these LHX motifs, you're on average further away from your promoter than if you don't have it, based on capture high-C data. Okay, so what about ZRS itself, right? So some of you might wonder, okay, ZRS somehow works. So when we looked at the ZRS sequence, and here I show you ZRS enhancer, and this is different binding sites for transcription factors that were characterized by other labs. And when we inspected the sequence of ZRS in more detail, we found that there are also 3 LHX sites that were previously uncharacterized, and those sites are conserved all the way to fish, right? So, okay, when we removed the sites, we tested, the first question is, okay, are they important for enhancer activity, right? And we tested it in a short range assay first, where we cloned it in front of the GFP. And so what we showed was that enhancer was still active, as you can see with this green signal here, right? So these motifs are not important for short range enhancer activity. However, when we remove those motifs at the endogenous location. And so this experiment took forever because we had to remove 3 different motifs at the endogenous enhancer. This resulted in a complete loss of sonic hedgehog function in the lamp, right? So this LHX, CTA, TTA motifs are critical for long-range activity of the ZRS, but not short-range activity. And to sum up what I just told you, so first, developmental enhancers are not always interchangeable at the remote endogenous location. If you swap a short-range enhancer at a long range, it won't work. It won't always work. We found an evolutionarily conserved signature consistent of this CA, TTA, LHX motifs that are specifically required for long-range enhancer activity, but not short-range activity. And so we found a prototypical range extender element that contains the signature that can extend the range of enhancers by more than 10-fold. So, and this is our current working model, right? So if you're a regular enhancer, you can work over short range, but you won't work over 400 kb. If you're an enhancer that has LHX motifs, it gives you an ability to work over short range and long range. And there is a special case when the Rex element containing this LHX motifs is located spatially separate from enhancer, in which case it gives an enhancer to act over long range. So I think the implications of this finding there are several implications. So first of all, I think it explains that the conundrum in the field where people move enhancers further away from the genes, and then the expression drops exponentially, right? And then at the same time, if you look at long-range developmental enhancers, they don't seem to care, right? SOX9 enhancers still work, right? ZRS still works. So there appears to be this is a pure speculation, there appears to be 2 types of enhancers in mammals. Those that are regular, they're short range, and enhancers that are long range, that have this Rex elements. And this long range enhancer can work over long range. And so there is some similarity between this Rex element and tethered elements that we discovered in flies, and also other facilitated elements. And the similarity is just conceptual, right? Because it's an element that is not an enhancer, but it assists enhancer in activation of the gene. And I think we'll find more of this type of elements in future. So, okay, in the last short part of my talk, I want to address the following problem. So, as I mentioned in the beginning, 90% of disease-linked variation maps to non-coding DNA, not the coding part of the gene. And many of this, so some of the variants, of course, disrupt the gene that are coding, but the non-coding variants, many of them affect enhancers, right? And it's very hard to study them because we don't know. We can't predict that, right? There's no computational tool. And then we don't know which gene is affected, right? And then in which cell type, right? And at the same time, there is a unmet need to actually test these variants because there is a lot of studies. This is just work from Matt Hurls and Sanger and others, where they sequence trios where a child has some disorder and the parents are unaffected, and they look for de novo variants, right? And then many of these variants are actually non-coding, but how do you actually test them, right? And as I told you, there's no computational tool to predict it. And you know, there are, of course, in vitro methods such as MPRA, but they're not available for most developmental and disease processes. And there is a need to test those variants in vivo in the context of development, especially for variants that cause congenital birth defects, right? And when I was a postdoc, we developed this method called unanswered, where it's essentially an enhancer reporter assay, but it's targeted, right? So it uses a self-harbor integration site in the mouse genome. So by that, we eliminate position effects. And so it's very efficient. More than 50% of injected eggs, more than 50% of mice are actually transgenic with a site-specific integration. It only takes 11 days. However, if you have an enhancer variant, let's say from some GWAS or whole genome sequencing study, comparing those enhanced activities is challenging. First of all, because LAG-Z is not really quantitative. And second, there is still some variability caused by mosaicism and other factors. So you have to, look at a lot of embryos. So MD PhD student in the lab, Ethan, had an idea. So why don't we put both enhancer and enhancer variant in the same construct, but separate them by insulator, right? And then one enhancer reference allele would drive red color, and the variant would drive green, right? And then we can just compare green and red in the same embryo, and then if everything is yellow, there is no effect. If there is an ectopic domain, some green activity, that's a gain of function. And if there is a loss of GFP, you'll see red, loss of function, right? So, and he spent a lot of time optimizing that method. We tested a lot of insulators, but in the end, he got it to work. And the advantages here is that it's very fast, right? So in 10 days, you get a readout. And so here's an example of a proof of principle, right? So this is the famous variant in the facial enhancer of IRF6 that is present in patients with cleft lip. When we put this variant allele and the reference allele in a dual insert, so we call this method dual insert, you can see that the embryos have only red activity driven by reference allele, but there's no green activity because it was killed by that variant, right? So in the same embryo, we can compare quantitatively. Another thing which is cool about this method is that it's a quantitative comparison, right? So it works. And then here's another example. So what I didn't tell you about ZRS before is that there are actually variants in that enhancer that cause polydactyly, and that is caused by ectopic Soren-Hedgehoff expression. And so when we put this variance in the ZRS and perform this dual reporter assay, you can see that the reference allele drives red only in this posterior domain, but the green, the GFP is active in the posterior domain and in this anterior domain that causes polydactyly, right? So again, so it also works for limb variants. And then we can also perform single cell experiments now from this dual fluorescent mice, and then identify cell populations where enhancer is misregulated, right? So in this case, this is a UMAP plot where you can see mCherry expression. And then if you look at the GFP expression that is driven by gain of function allele, you can see this ectopic cell population shown here, right? And you can characterize gene expression there. Okay, so this was known variants, right? So it's already something we knew before. Of course, it's nice to have a proof of principle. So can we actually use this method to screen previously uncharacterized variants? And to do that, Ethan collected all the variants from whole genome sequencing and GWAS studies from patients with neurodevelopmental disorders. He identified thousands of those variants and then looked at the variants that were within experimental validated enhancers. And then we tested them using this dual insert. And in doing so, we found some very interesting examples. For example, here, there are two variants from independent patients with autism that fall in this brain enhancer of microRNA-9/2. And when we do dual insert, what we see is that these two variants kill enhancer activity completely, right? So there is no green at all, right? And interestingly, variants fall within predicted transcription factor binding sites. And so here's another example, which is very interesting. It's a common variant, which is actually a short tandem repeat in the enhancer of OTX2. And that variant causes gain of activity in the midbrain, as you can see here in the green. And then you can quantify it. So that shows that we can identify and functionally test variants from patients and see where exactly and when enhancer is affected. So to summarize here, I presented you this dual insert method that allows you to quantitatively compare enhancer variant activities in the same mouse in just under 2 weeks. And then we can assess both loss and gain of function variants and both rare and common variants that we showed, and we can quantitatively compare them. And we can also combine it with single-cell RNA-seq for more in-depth characterization of cell types in which enhancer is misregulated. And so I mean, our hope here is that it will be useful for human geneticists when they identify those thousands of variants and then prioritize them with MPRA. And then this could be like an orthogonal approach to test non-coding variants in vivo, which could be especially powerful for congenital abnormalities that happen during development. So I want to thank my fabulous lab. And so these 3 projects that I talked about were led by 2 very talented grad students, Grace and Ethan, and 1 very talented postdoc, Joashin. She will be soon on the job market, and our collaborators and funding. And I'm looking forward for discussion.